Good morning, all. Let's take our seats, settle down on this rainy day. Okay, so now we're going to be doing connective tissue and cartilage today, and then on Monday, bone development. And all four of these lectures are more or less in a continuum. They relate to each other and they build upon each other, and hopefully you'll see how that works as we go along. So if you're thinking of being an orthopedist, this is the lectures for you. So here are a couple of learning objectives. So firstly, connective tissue. So we need to classify connective tissue, and we'll go over that momentarily. And there are cellular components of connective tissue, and there are extracellular components of connective tissue that are secreted by those connective tissue cells. And of the extracellular, well, you need to know the cellular components and also the extracellular components, be able to describe and identify what we call amorphous and fibrous components, and know the differences between those different types of extracellular connective tissue. Okay? Let me give you the big picture in connective tissue. Okay, the big picture. So way back in the beginning, well, firstly, what I want to say is the evolutionary sequence in phylogeny, i.e. the evolutionary development of connective tissue, we see that repeated somewhat in embryological development, i.e. in ontogeny, and significantly that repetition we see in bone repair. Okay, when you break a bone, you go through a sequence of firstly deposition of fibrous and amorphous components, i.e. non-cellular, extracellular components. Then there's a cartilage bridge, and then we get bone. And that's what we see during evolution. We see that sequence of fibrous, amorphous components, cartilage and bone. So way back when, with our prokaryotes, remember our prokaryotic story, their prokaryotes, like bacteria, already express a proto-collagen gene or genes. We don't know what they do in bacteria, but they're there, which is very fortuitous, because then they're available to evolve into something more useful in metazoan life forms, i.e. metazoans being multicellular life forms. And of course you can imagine to have a multicellular organism, you've got to stick the cells together, right? And one of the things that you can use to stick the cells together with is connective tissue. It connects tissues, connects cells. It makes sense. And that connective tissue can be either fibrous or amorphous. And we think maybe that evolved in the Precambrian, maybe 850 million years ago or something or other like that. We don't know really quite clearly, because fossils are exceedingly rare from that period. Then if you take these fibrous and amorphous components, and if you have the right combination of specifically amorphous components, you can make cartilage, which is a lot more rigid, and that's very useful. Here's a picture of hyaline cartilage. This little, here he is. It's hard to see this little guy. The hyaline cartilage here has a lot of amorphous components, but it's a little more rigid. It's flexible, okay, and that's useful because then you can attach things to it, like muscles, things like that, so you can make more complex organisms. And if you take that cartilage and then you calcify it, you can make bone, okay? So we see this sequence during evolution from the proteins to make metazoans, then cartilage, then bone, and we see that sequence repeated in embryological development, and we see it repeated during bone repair. So in effect, all three of those events or phenomena are interrelated, okay? They're interdependent. You can see logically how bone repair occurs, because ontogeny follows phylogeny, and that is repeated during bone repair. And we will see that much more so next Monday when I talk about bone repair in greater detail. So here's some early metazoan life forms, okay, really primitive. Who knows what these things are, okay? Going back at least 650 million years in the Precambrian. You can start to see some body segmentation. This looks more like something you might recognize, a head, maybe a notochord and some sort of structures like that. But obviously to get this sort of organism, we needed connective tissue to hold the different cells together. Okay, so connective tissue. We can classify connective tissue in a number of different ways. This is a man-made classification. So connective tissue proper, we can have loose connective tissue. A good example of that is the lamina propria, you know, those cells and extracellular matrix materials that underlie most epithelial sheets. So that's loose connective tissue. Or we can have dense connective tissue. And dense connective tissue can be either regular or irregular. So dense regular connective tissue, examples are ligaments and tendons. We'll look at those more so as we go along. Or irregular dense connective tissue such as dermis. Okay? So when we talk about dense, we're talking about the compaction of the cells and the extracellular material. And if those cells and extracellular material is arranged very evenly, like in nice long orientations, similar orientations, that would be regular. And if it's more jumbled around, you know, in different orientations, that would be irregular. So, okay, so dense connective tissue, we have regular and irregular. There's no regular or irregular loose connective tissue. It's all irregular, okay? So we don't classify loose connective tissue as either regular or irregular, because it's all loose. It's all irregular. Then we have connective tissue with special properties, shown here. Maybe I should, shown here, okay? And then one type is adipose tissue, which are fat cells, okay? And we'll be looking at that momentarily. We also have elastic tissue, which has elastic fibers in there, which is by the name implies it has elasticity, and we'll be looking at that in some greater detail. Hematopoietic tissue, that's basically the components of blood, okay, and we consider the blood to be connective tissue because the components of blood are circulating throughout the body, so they're connecting and communicating with one part of the body to another, i.e. whether it's the origin of those cells mostly in bone marrow and then carrying out a function elsewhere, or an infection. Maybe you've got some lymphocytes that are stimulated by some infection elsewhere. So because of the blood is circulating, we consider the blood to be a form of connective tissue. And then we have mucous tissue, shown here. Here they're talking about mucosa. So mucosa is a generic term that basically means the lining of many of the tracts of the body, like the GI tract, the respiratory tract, reproductive tracts. Basically, that's the lining epithelium, and the underlying, usually the lamina propria. Okay. Then we have connective tissue with special properties such as cartilage. I kind of briefly mentioned that, and bone. And both of these play a supportive role because they are much more rigid and provide for a skeleton, if you like, upon which you can attach muscles and things like that. So that's our classification of connective tissue. Let's look at some of these. So basically, pretty much all connective tissue has a embryological origin of mesenchyme, which looks like this. It's pretty nondescript. And then it will differentiate into more recognizable cell types. So here we have our two origins of connective tissue. The first is the hematopoietic connective tissue, and it gives rise, so hematopoietic meaning from the blood, from the bone marrow. In the bone marrow, there's these stem cells that give rise to all of these cells. So it gives rise to what we call granulocytes, because they have granulocytes in the cytoplasm, neutrophils, eosinophils, basophils, and mast cells related to basophils. Okay, we will look at that in great detail when we do the components of the blood and the origin of the cells. Cells in the bone marrow give rise to megakaryocytes, which then break up to give platelets, which play a role in blood clotting. They also give rise to lymphocytes. There's there's several different types of it. The main types are B cells and T cells. B cells differentiate into plasma cells and produce antibodies. Monocytes, okay, they differentiate into macrophages, which are phagocytic, And monocytes can also differentiate into similarly related cells that have a phagocytic function, such as Langerhans cells in the skin, microglia of the brain or the nervous system, osteoclasts of bone, which we will be discussing next week. Okay, so all of these different components then are derived from hematopoietic stem cells in the bone marrow. The other origin of connective tissue cells is the undifferentiated mesenchymal cells that I showed you in that previous slide. They can give rise to fibroblasts. Fibroblasts secrete a lot of the extracellular material that we'll be discussing, either fibrous or amorphous. We also have fat cells, which act as the storage of energy. Chondrocytes and chondroblasts are cellular components of cartilage. Osteoblasts and osteocytes are cellular components of bone. We also have smooth muscle, which surrounds blood vessels, and the endothelial cells that lines the blood vessels. And mesothelial cells is a type of epithelial cell that lines many of the internal cavities, like the abdominal cavity, things like that. Okay, so then we've got basically this bifurcation for the origin of connective tissue cells. We've got the hematopoietic cells derived from bone marrow, and the mesenchymal cells, which are basically embryologically undifferentiated cells that then differentiate into these other cell types that I just described. Okay, here are some of the, how do we get rid of these dots? Is there an eraser? They should, okay. It's supposed to be, oh, there you go, great, thank you. Okay, so here are some of the cell types we've been talking about. Fibroblasts, chondroblasts, osteoblasts, odontoblasts, they're all structural. Okay, chondroblasts, cartilage, osteoblasts of bone, odontoblasts of teeth. And they produce fibrous material, plays a structural role. Plasma cells derived from B lymphocytes will produce antibodies, so that's an immunological role. Lymphocytes, B cells can differentiate into plasma cells, but T cells are a type of lymphocyte that can participate in cytotoxic T cell killing. It can directly attach to a foreign pathogen and destroy it. Eosinophils contain granules that play a role in destruction of parasitic infections like helminthic worms and things like that, so that's also defensive. Neutrophils, most common cell type, nucleated cell type in the blood, has a phagocytic role, major role in destruction of bacteria. Macrophages derived from monocytes obviously can phagocytose foreign pathogens such as bacteria, and they can differentiate into some of the other related so-called antigen-presenting cells, such as the Kupffer cells, osteoclasts, microglia, and so on. Mast cells and basophils play a role in inflammatory response. So when you get bitten by a bee or a mosquito and you get that little inflammation, that bump, or some people have severe inflammatory responses, like when you have if you have a peanut allergy, things like that. These are the cells, the mast cells and the basophils, that are mediating an inflammatory response. And then the last cell type is the adipose cell, the fat cell, which acts as a reservoir of energy. Okay, so components of connective tissue. So we can divide the components of connective tissue into a cellular type, and then there's the extracellular material. So the extracellular material is the stuff secreted by the cells, okay? And we can further divide the extracellular matrix into amorphous, meaning under the microscope we really can't see any sort of distinct structure, or fibrous, which has a fibrous, clearly defined fibrous type of structure that we can see under a microscope. So if you first look at the cellular components, we have fibroblasts, adipocytes or fat cells, the macrophages, which are phagocytic, the mast cells, inflammatory response. Leukocytes is a term meaning the white blood cells in the blood, which includes all the granulocytes, the monocytes, the lymphocytes, and so on. And plasma cells are derived from B lymphocytes, which secrete antibodies. Okay, so here are fibroblasts, the top panel being the light microscopic view, the bottom panel being the electron microscopic view. Kind of nondescript. You can see a nucleus, a bit of cytoplasm. The thing to note is that fibroblasts are secreting a lot of extracellular material. Okay, so there's going to be some collagen fibers and some amorphous material being secreted by the fibroblasts, and they're pretty much distributed throughout the body, and they can undergo proliferation as required to deposit extracellular matrix and play a role, structural role in sticking things together. Then we have adipocytes or fat cells. So this whole thing is a fat cell. This is a scanning electron micrograph, and this is what we call a unilocular adipocyte. Uni meaning there's one big lipid droplet inside this fat cell. Here's the light microscopic view. You can see this whole cell is filled with one lipid droplet. Now this on the edge, this is not just plasma membrane. This is also the cytoplasm that's been squeezed out to the periphery. You can see some nuclei here and here and here. Okay, so we call this a unilocular adipocyte because there's one huge lipid droplet that fills the entire cytoplasm of the cells. This is the fat cells that you and I have in our body currently. Then we also have a multilocular adipocyte. We don't have those anymore. Multilocular meaning many lipid droplets. So here's one, here's 2, 3, 4, 5, 6, 7, at least 7. This is the nucleus in the center, right here. And also, the multilocular adipocytes have many mitochondria. See these many different mitochondria here and here and here. So multilocular adipocytes are also called brown fat. Okay, some animals have retained brown fat. Babies have brown fat when they're born. It usually disappears after about 3 months. So you did have it once, but not anymore. The thing about multilocular adipocytes is that because you have these smaller vesicles, you have a higher surface area to volume ratio. Have you ever done that stuff where you take a structure and you do a ratio of surface area to volume? So as something shrinks in size, the ratio of surface area to volume actually increases. The surface area is bigger now compared to the volume. So that makes that more accessible for metabolism because you've got an increased surface area to volume ratio of a smaller droplet. Okay, so another way to think about it is surface area to volume ratio. You've got a loaf of bread. Okay, let's just say the loaf of bread has a unit surface area to volume ratio of 1. If you slice that bread, okay, it's the same volume, but you've increased the surface area because you've got those additional surface areas of every slice. So by simply making the slice of bread, by slicing it, you've increased the surface area to volume ratio. Volume's the same, but the surface area is increased because you've cut it into smaller slices. So that's kind of the concept. So you have an increased surface area to volume ratio because you have smaller lipid droplets, which makes them more accessible for metabolism. And these mitochondria are right there to metabolize that lipid. So basically, this is a way for the adipocytes to rapidly generate heat, okay, in newborn babies. So if ever you've seen a film or maybe some relative of a baby being born, has anybody ever seen a baby being born? Film or in life? Okay, the first thing they do is start squawking, but the other thing they start doing is shivering, right? It's an instinctive thing. It's an evolutionary instinctive thing for survival. I mean, they're in a nice hospital these days where the temperature's controlled, but they're shivering anyway. And that's an evolutionary instinctive thing because a couple of hundred years ago, we didn't have nice air-conditioned hospitals. You know, babies were born in a hut somewhere or under a tree, in a cave, couldn't control the environment. So then, so who knows what the temperature would be. So shivering is a mechanism for them to survive those early times after they're born, because then you can get swaddled up and then they're okay. So what they're doing is when they start shivering immediately after they're being born is they're burning the fat, the multilocular adipocyte fat, to generate heat as a survival mechanism. Now some animals have retained, I think bats actually have a lot of brown fat for some reason. But in humans, the brown fat, the multilocular adipocytes, disappears after a couple of months. Here's another picture of brown fat. You can note here then that we have multiple lipid droplets. So these are the multiple lipid droplets. We have multiple mitochondria, although you really can't make them out too clearly this light microscopic picture. And of course, as I mentioned, the combination of smaller lipid droplets and abundant mitochondria makes more readily available to generate heat in newborn babies. Okay, next cell type are macrophages. These are phagocytic in function. So here's one macrophage here, here's one here, here's another one here. Okay, now how do we know that these cells are phagocytic? Well, what's happened is that these cells, these macrophages, have, in this particular animal, probably a mouse or a rat, have been injected with Indian ink, which is this black lipid, these black droplets that you see, and these phagocytic cells have phagocytosed the Indian ink from this organism. And so we can readily see that the function, this is a functional demonstration, that these macrophages have a large capacity for phagocytosis. And of course that's useful because one of the things that they may phagocytose is bacteria, okay, if you have an infection, or other things, maybe virus, things like that, any sort of pathogen, they're going to phagocytose it. And later on we'll learn about the role of macrophages in stimulating the immune response. But for today's lecture, all we need to really take in is that macrophages have a phagocytic function. Okay, the next cell type are plasma cells. Plasma cells are derived from B lymphocytes. If the B lymphocytes are stimulated, they differentiate into plasma cells and produce antibodies, okay? And the characteristics of plasma cells is that they have an eccentric nucleus, meaning off-center, right? So this nucleus is off-center from the cytoplasm. They have a cartwheel arrangement of heterochromatin, so this might be the axle, and then you've got spokes like here, cartwheel. And also in an electron micrograph, we can see that they have abundant rough endoplasmic reticulum, okay? So RER down here. So again, here are the characteristics. You can see the eccentric nucleus, it's off-center, cartwheel arrangement of the heterochromatin, okay, and the abundant RER, okay, here. And of course, we have an abundance of rough endoplasmic reticulum because the function of these cells is to produce immunoglobulins, antibodies. So you need a lot of rough endoplasmic reticulum to produce the antibodies to fight a foreign pathogen. The next cell type are mast cells. This is an electron micrograph of a mast cell. Mast cells play a role in inflammatory response, okay? And they contain these multiple granules. You can see these large dark granules in this electron micrograph of mast cells. And mast cells then, as I said, play a role in inflammatory response through the activation of immunoglobulin E receptors. There's different types of IgG, immunoglobulins. IgG you're probably familiar with, but there's also IgM, A, D, and E, which are the 5 common types. And subtypes within there, but we're not going to get into that. But IgE is a low abundance immunoglobulin that is bound to the surface of mast cells via a receptor. And if you just say you are bitten by a bee and that antigen, B antigen, makes its way into your body, that B antigen then can cross-link to adjacent IgE molecules. So when those two adjacent IgE molecules come together, that initiates signal transduction inside the cell, because now these intracellular proteins that are associated with the receptor there can then come together and initiate a signaling event shown here. And as part of that signaling event, you get mobilization of calcium, okay, which can cause, in some cases, the fusion of granules together to make larger granules, and then eventually fusion of those granules with the plasma membrane, releasing the contents of those granules, which include heparin, which is a anticoagulant, right? Histamine, which is a vasodilator, some proteoglycans, which is a form of amorphous connective tissue. And then there's also an eosinophil chemotactic factor, which will stimulate the recruitment of eosinophils from the blood to that site of inflammation. So then the bottom line here is that mast cells and basophils having a very common function together promote an inflammatory response, such as swelling. When I say inflammation, I'm talking about a swelling, localized edema, right? So like a bee sting or a mosquito bite, you get a bump, that's localized edema, fluid flowing into that site caused by, in part, these cells. So here are some of these cell types that we just looked at. Most of what you see there are the red blood cells that don't have a nucleus. We can see a lymphocyte. Let's see a lymphocyte here, a monocyte here, and these are neutrophils up here. Okay, and these are platelets, and of course these are red blood cells. Okay, so that's the cellular components and the functions of the cellular components. Let's look at the extracellular components. This is the stuff secreted by many of those cells. And the first component that I want to look at is the amorphous component. So the amorphous component of extracellular matrix is amorphous, as the name implies. It doesn't have a form or a body. In other words, it's kind of a fuzzy, vague type of structure that you really can't make out any definitive structure. With the light microscopic view, you need high-power electron microscopes to really see anything. And the amorphous components come in three different types, classifications. There are glycosaminoglycans, which are basically disaccharide amino-containing sugar residues linked in a long linear chain. So basically they're sugars. Then there's proteoglycans, which are a combination of proteins and the glycan being glycosaminoglycans attached. And glycoproteins, which is mostly protein with some disaccharide residues which are branched. So the glycosaminoglycans are unbranched, they're long linear chains. The glyco component of the glycoproteins, typically the saccharides are branched. So we go from pretty much all sugars, 50-50, to mostly protein with a little bit of sugar in here. So there's a gradation of all sugar down to lesser sugar and mostly protein up to no protein in that particular arrangement. Okay, so a large glycosaminoglycan that we see quite often is hyaluronic acid, shown here. It can be more than 1 million Daltons in size, and as I said, it's an unbranched polymeric disaccharide containing amino sugars. So what does that mean? So disaccharide, so here's one sugar residue, two sugar residues, so two disaccharide. So that's the disaccharide, if you like, a doublet of sugars. One of them is an amino containing sugar right here, okay? So that's our disaccharide containing amino sugars. And if you form a long linear chain of this subunit, so this is one of the subunits, N, so you make a polymeric chain of these disaccharide repeating units. Polymeric meaning multiples of, hundreds, thousands of units linked together in a long linear chain. So that's what glycosaminoglycans are. And so then proteoglycans is a combination of protein and glycosaminoglycans. So in this particular case, we can see there's a glycosaminoglycan down the center, and then branched off is a protein, okay, link shown here, core protein. And then off that core protein, we can have other glycosaminoglycans which form these sort of bristles, if you like, off the edge of the protein. So then a proteoglycan is a combination of protein and glycosaminoglycans. This is shown here. So this bottle brush structure up here is a proteoglycan. So we have the protein in the middle, and we have the glycosaminoglycans on the side, okay? So that's proteoglycan. Whereas down the bottom we have a glycoprotein. So the pretzel that we see is the proteinaceous component, and the glyco up here is the branched saccharide units, the branched sugars, okay? So in the glycoprotein, the sugars are branched, whereas you remember in the glycosaminoglycan, they're unbranched. It's a long linear chain. So glycoproteins, there are different varieties, but we see some that are common in connective tissue, such as fibronectin and laminin. The thing to note about these connective tissue proteins is that they have sequences that will bind other components of connective tissue. So we have a collagen binding sequence, a cell binding sequence, a heparin binding sequence, proteoglycan binding sequence, collagen binding sequence, and so on. So basically these binding sequences of fibronectin and laminin will bind other different types of extracellular matrix and link them together. Hence the term connective tissue. They're connecting one of the various types of connective tissue extracellular matrix to another type, forming, if you like, a mesh-like connection between different types of proteins and also to cells. So, and the way that these connective tissue components are linked to cells are through integrins. So integrins are a family of proteins. Actually, they're dimers consisting of an alpha and a beta subunit, and there's different types of alpha and beta subunits. So you might have alpha 1 through 10, beta 1 through 12, something like that. So the combination of different alpha and beta subunits specifies what they will bind to. So for instance, I'm just making this up, say an alpha 3 beta 2 might bind type 1 collagen, whereas a alpha 5 beta 1 might bind some sort of glycosaminoglycan. So by having the appropriate combination of alpha and beta subunits, you can bind specific types of extracellular components of connective tissue. Now these integrin proteins then pass through the cell membrane and they're linked to intracellular cytoskeletal components. So then they form a bridge which will link the cell bound to extracellular material. So it's holding the cell in place, if you like, hence connective tissue. It's connecting the cell to the extracellular material via these integrins. And you may recall from the cell organelles 2 lecture that in terms of cell junctions, hemidesmosomes connect a cell to the basement membrane. In that case, cadherins are replaced with integrins to hold a hemidesmosome to the extracellular components of the basement membrane. Okay, so that's the amorphous components, which means the stuff we really can't see very clearly, is kind of fuzzy. And then the other classification of extracellular material is the fibrous component. The fibrous components of connective tissue come in 3 categories. There are the collagen fibers, there are reticular fibers, which as it turns out are really just type 3 collagen, but when they first classified these structures they thought there was something uniquely distinct, but they're not, they're type 3 collagen, and I'll go over that again. And the other major gene product of classification are elastic fibers, which are completely different to the collagens. Okay, so collagens, there are more than 20 collagen types, there's a lot, okay? Some of them are kind of common, 1 through 4. So in the beginning, the collagens were a structural protein, and it evolved and became diversified in function. And as I mentioned, even in bacteria, we have protocollagens. So this was our protocollagen genes. This was our starting point in bacteria. Function unknown, but it was available for evolution into true collagen fibers, which can then be used as a connective tissue component. So because they were present in such primitive organisms, all phyla of the animal kingdom contain collagen. So the most primitive of the animals of the animal kingdom, such as sponges or jellyfish, even they have connective tissue because everything above them. They're the most primitive organisms, other than unicellular organisms, but of multicellular organisms. So collagens then do a bunch of things. Adhesion, of course, they're sticking cells and tissues together. Skeletal function, holding things together. They're very present, very prominent in the dermis, which is basically what leather is, the dermis of the skin, and they can also do some signaling. So let's look then at the synthesis of collagen fibers. This schematic gives you a generalized overview of how collagen fibers, all of the different types, are synthesized, say, in a fibroblast. So we start out with the synthesis of a pre-procollagen molecule. So the pre is the signal peptide, okay, and that is clipped off. The signal peptide is clipped off as that pre-procollagen molecule passes from the ribosomes into the lumen of the endoplasmic reticulum. So refresh your memory with the synthesis of proteins, and we talked about how proteins are synthesized in or on rough endoplasmic reticulum and then make their way into the lumen, right? So that's what we're talking about here. So as that pre-procollagen molecule makes its way into the lumen of the rough endoplasmic reticulum, a small component is clipped off. That's the signal peptide. So once our signal peptide is clipped off, now what is the procollagen molecule is in the lumen of the rough endoplasmic reticulum, where it becomes hydroxylated, okay? And that hydroxylation occurs in the presence of vitamin C, which acts as a cofactor. So if you have a deficiency of vitamin C, you have scurvy. I'm sure you've all heard of scurvy. And that scurvy is caused by a deficiency of vitamin C, which then leads to a functional absence, if you like, of hydroxylation of collagen molecules. Okay, so once we get hydroxylation, we also get some glycosylation. Okay, remember there's initial glycosylation of proteins in the lumen of the endoplasmic reticulum. And then our procollagen molecules are assembled as a procollagen molecule into a triple helix. So here we can see our triple helix. And this arrangement into a triple helix occurs by virtue of this squiggly line registration peptide. So the registration peptide does two major things. It allows for alignment of the triple helix, aligns them up. Here you've got three molecules, it aligns them up into a triple helix. And also it maintains that procollagen triple helix molecule in a soluble state. Then as that procollagen molecule is secreted into the extracellular environment, it comes into contact with an enzyme, a procollagen peptidase, that clips off a chunk of the registration peptide. So once you remove a chunk of the registration peptide, you've changed the physicochemical characteristics of that triple helix. Okay, so it is no longer procollagen. Now that you've chopped off a piece of it, it is now tropocollagen, and that change in physicochemical characteristics causes that now what is we're calling tropocollagen to precipitate out as a microfibril. Okay, so you cut off a piece of procollagen, cut off a piece of the registration peptide, it changes its physicochemical characteristics, precipitates out as a fibrous protein. And then that tropocollagen microfibril can be bundled together into, small fibers, fiber bundles, and so on. So here are our collagen fibers shown here with this arrow in red. You can see how thick it is relative to the elastic fibers, which are much more narrow with the black line on top up there. In electron micrographs, we can see collagen fibers. Depending upon the type of collagen, we can see in longitudinal section these striations, or here it is in cross-section. Okay. And we have different types of collagens that, if we have a functional deficit in them, that can give rise to various diseases. So here's just a couple of different diseases that can lead to weak, so you can have, defects in type 4, type 6, type 7 collagens. So they're given different names. And these obviously lead to weakened connective tissue, and you can have various organs and tissues that are affected. Also scurvy, I talked about a lack of vitamin C will give rise to scurvy. So here, this is what scurvy can look like. Okay, scurvy results from a lack of vitamin C. So you've got weak connective tissue. So a major component of the dermis of the skin is the connective tissue component. So you're going to have ulceration. The skin is weak, it's breaking, it easily breaks. You can see here these ulcerations, teeth are falling out, right? So obviously scurvy is not very good in terms of if you want to be able to do physical labor. So historically, you may recall during the discovery of the Americas, from Europe, these sailing ships would be coming across the ocean. They'd take them about 2 months to get from Europe to the Americas, and during that time they had no fresh fruits and vegetables, they had no vitamin C, they'd be eating salted fish, stuff like that. So by the time they got to the Americas, a lot of those guys had scurvy, right? The sailors, because they didn't have access to fresh fruits and vegetables. So a lot of the time they found that if they could stop off at an island somewhere where there was some fruit, then they could give the sailors fruit, okay, and then that would counteract the scurvy, and then the sailors would be more physically able to carry out their functions. So that's how some of the islands of the Caribbean were discovered. The captain of the ship was not just looking to find gold on these new worlds, but he was also looking for a source of vitamin C for his sailors, okay? Okay, here's another disease, Ehlers-Danlos syndrome, another defect in collagen synthesis. You can see how this person can bend their hands way back, right? Because the collagen is so weak, okay? They've got this condition. Okay, interesting fact. Turns out about a decade ago, maybe longer, some scientists extracted the contents of the bone marrow of a T-Rex, a dinosaur, right, which was 68 million years, and they took that contents of the dinosaur's bone marrow cavity, and they sequenced it, and they found that they were able to actually sequence collagen from this 68 million year old dinosaur, which is pretty amazing. And they've done it with other dinosaurs. It was very controversial at the time. People didn't believe it, but they've done it with other dinosaur bones going back more than 110 million years, and they can sequence collagen fibers. So when they sequenced the collagen fibers from this dinosaur, T-Rex, they found that it was most closely related to modern-day chicken collagen fibers, okay, which makes sense because we didn't have many mammals 68 million years ago. Hardly any, they were still trying to survive. And of course, birds evolved from dinosaurs. So it makes sense that the collagen fibers of a dinosaur T-Rex would be most related to modern-day chickens. But most significantly, and the point of this slide is, the fact that collagen fibers can survive for 68 million years tells you how resilient this component of connective tissue is to degradation, because that's one of the things that connective tissue has to do. It has to be very resilient to degradation during a lot of bending and holding of things together. So collagen fibers are very resilient to breakdown. They're slow to be synthesized, but slow to break down. So this just kind of highlights that fact. Okay, so has anybody seen the movie Jurassic Park? 1, 2, 3, right? So just imagine it's real, and you go to the island for Jurassic Park, and you remember that cafeteria where those little kids were being chased around by the velociraptors, they're hiding in the cupboards of the cafeteria. So you go to Jurassic Park and you go to the cafeteria, and you have lunch at the cafeteria, and they've got dinosaur steak on the menu. So you have dinosaur steak, you go home, tell your friends, hey guys, guess what? I had dinosaur steak at Jurassic Park. And they say, yeah, what does dinosaur steak taste like? And you know what the answer would be? Tastes like chicken, right? There you go. Okay, so much for collagen fibers. Elastic fibers. Elastic fibers are completely different to the collagens, different gene products. And the evolution of elastic fibers is much more recent. They are present in jawed vertebrates, but not present in jawless vertebrates like lampreys, okay, which is kind of interesting. And it turns out that elastic fibers first appear in these jawed vertebrates. It's thought in response to these jawed vertebrates having a high pressure closed circulatory system. So these more primitive jawless vertebrates don't have a particularly high pressure circulatory system, but elastic fibers are in these more evolved vertebrates, which have higher blood pressure, and because the blood vessels are expanding, contracting, expanding, contracting, you need some elasticity, and that's where you see a lot of elastic fibers in blood vessels. So actually, it's only 400 million years ago they evolved, which is pretty recent when you think about how far back we go and when things have evolved. So elastic fibers are actually made of two different gene products. We start out with what are sometimes called oxytalin fibers, which is made up of fibrillin. So you see all these fibers. These are the fibrillin fibers. And we see them completely by themselves as zonal fibers in the eye connecting the ciliary body to the lens. When I give you this eye lecture in November, I'll remind you that these zonal fibers are actually oxytalin fibers as shown here. Then, if you have these oxytalin fibers, a different gene product can be synthesized and secreted, and this is the elastin. So you can get little blobs of elastin that starts to fill in around the oxytalin fibers, as shown here, and then we see this arrangement, which is called elaunin, in and around the sweat glands. And then if you keep secreting this elastin gene product, eventually you get a big blob which completely fills in the oxytalin fibers, and then we get what is called a elastic fiber. Now this actually is reminiscent of what we see in structural engineering. So for instance, when you see a building or a sidewalk or something where they're pouring concrete, the first thing that they lay down is the rebar, right? You see those steel rods, either it's the frame of a pillar, or if it's the pavement, it's, you know, crosses throughout. So think of that rebar as the fibrillin fibers, and the cement is the elastin. So this arrangement of the fibrillin and the elastin, or the rebar and the cement, gives structural integrity to that structure. You can stress it and stretch it, and it's not just going to crumble. If you had just a piece of concrete all by itself and you whack it, it's going to break. But having the rebar in there holds it together, and it's much more resilient to degradation. Same thing with elastic fibers. The fibrillin renders some sort of structural integrity so that the elastin won't break so easily. So evolution came up with a solution long before structural engineers did in terms of pouring concrete. So here's our elastic fibers up the top there, little thin black fiber. Sometimes we stain elastic fibers with orsine. Okay, that's a common stain for elastic fibers. And once that disappears. Okay. As you can see, the size of the elastic fiber compared to the collagen fiber, which is shown with the arrow, which is much thicker. Okay, and then of course the thing about elastic fibers is that you can stretch them and relax them, and they will retain their structural integrity. Okay, they won't break apart, which is very important because elastic fibers are localized in regions of the body where you have a lot of stretching, such as blood vessels, places like that. So here's a disease that can occur due to a mutation in the fibrillin gene. So remember, fibrillin is the oxytalin fibers, the first part that's deposited during the formation of elastic fibers. So if you have defective elastic fibers, this person that has Marfan syndrome can have an aneurysm of the aorta. So in other words, when the aorta expands during the pumping of blood from the heart, it's not contracting back completely fully, and they have this aneurysm, kind of like a bulbous blowout, like a tire blowout on a bicycle. It pops out, and then eventually that may rupture, that aneurysm, and that person will bleed to death. So this is actually a congenital disease. Sometimes they don't even know they've got it until something happens and they die. Okay, Marfan syndrome, but that is directly due to a mutation in the fibrillin gene, the first component of the synthesis and deposition of elastic fibers. Okay, then the last component of the fibrous component of connective tissue I want to talk about are the reticular fibers. Now, we used to think this was totally separate from the collagens and the elastic fibers. It turns out it is simply type 3 collagen. Okay, and we see reticular fibers enriched in lymphoid organs and tissues, okay, such as lymph nodes, spleen, thymus, places like that. And we can stain reticular fibers with a silver-based stain, and it stains black, so it says arginophilic, argir, silver. Okay, when silver oxidizes, it becomes black, right? So arginophilic, silver-based stain. So here we can see some reticular fibers in a lymph node, where the arrows are pointing to blackish, I guess there's a little more purple on this one, reticular fibers within the lymph node. And we can compare reticular fibers here in cross-section with type 1 collagen. Remember that reticular fibers are type 3 collagen, and this is type 1 collagen. And you can see the diameter in cross-section of the reticular fibers is perhaps 30 nanometers as opposed to the diameter of the type 3 collagens, which we'll say is about 70 based upon this histogram here. Okay, but they're both collagen fibers, different diameter, different genes. Okay, then the last thing I want to talk about is how the connective tissue of the extracellular environment plays a role in controlling the flow of fluid into and out of the smallest of the blood vessels, the capillaries. So when we have blood coming from the heart, it has a high blood pressure, and as it makes its way through the various blood vessels, the blood pressure declines because you've got an increased volume of the blood pressure, and also it's rubbing against the wall, so you've got a slowing down of the blood vessels, the blood pressure in the blood vessels, and we call that hydrostatic pressure. The pressure of blood coming out of the heart is the hydrostatic pressure. It's the same as water coming out of a tap. That's hydrostatic pressure. And that declines from the heart down to the smallest blood vessels. Conversely, in the connective tissue space outside of cells, we have the plasma colloidal pressure, which is the osmotic pressure of that connective tissue fluid due to the various proteins and extracellular matrix components, glycosaminoglycans and so on, in that tissue. And that is relatively constant. So the colloidal osmotic pressure outside of cells in the tissue space is constant at around 25 millimeters of mercury. So you can imagine on the arterial side of blood vessels, the hydrostatic pressure exceeds the osmotic pressure, so fluid is going to flow out of arterioles into the connective tissue space. Then beyond the capillaries, on the venous side, the blood pressure has dropped so low in the veins and the venules that now the osmotic pressure of the tissue space exceeds the blood pressure in the veins and the venules. So then fluid flows from the connective tissue space back into the venules, whereupon it makes its way to the heart. So the simple difference in pressure, hydrostatic pressure in the blood vessels, constant osmotic pressure of the tissue space determines the flow of fluid either out of the arterioles and capillaries into the connective tissue, or on the venous side of things, the flow of fluids from the connective tissue space back into capillaries and venules for return to the heart. Okay, so it's a simple difference in physics, if you like, of pressure between those two spaces, the cardiovascular space versus the connective tissue space. This is shown again here schematically. On the arterial side of things, hydrostatic pressure, arterial side of things, arterial pressure, the hydrostatic pressure of arterioles exceeds the osmotic pressure, so then fluid's going to flow out into the connective tissue space. Conversely, on the venule side of things, the osmotic pressure here exceeds the hydrostatic pressure in the blood vessel, so then fluid is going to flow back into the venule. So flows out of the arterial end, flows back in to the venule end. So of course on the arterial end it's carrying oxygen and nutrients. On the venule end the oxygen's been used up, now you've got carbon dioxide and metabolites which you want to return to other parts of the body for processing. So that's why we have that. Okay. Here is our question for lecture number 1, connective tissue. Which of the following is derived from hematopoietic stem cells? Go back to one of those first slides I showed you. Hematopoietic stem cells. Which one of these different cellular components of connective tissue is a hematopoietic stem cell derivative as opposed to the mesenchymal cell derivative? Okay? Osteoclasts. There's our answer. Okay, let's take a break. And on the half hour at 11:30, we'll start with the cartilage lecture. Okay, so now we know the cellular and extracellular components of connective tissue. And the extracellular components can be either amorphous, glycosaminoglycans, proteoglycans, or glycoproteins, or they can be fibrous. The elastic fibers, reticular fibers, which is basically, collagen fibers, reticular fibers, which is type 3 collagen, or the elastic fibers. So now we can arrange those different components to make cartilage. So now we're going to talk about cartilage. So things that you should hopefully learn: know the cellular components of cartilage and the extracellular components. There are distinct amorphous and fibrous components that make up different types of cartilage. You can know the different types of cartilage growth, and know the locations of interstitial growth, which is one of the less common types of cartilage growth, and then know some of the distinguishing characteristics of the three histological classifications of cartilage: hyaline, elastic, and fibrocartilage, amongst other things. So where do we find cartilage? Well, in the adult, it's kind of restricted to a number of different locations. We see it in the ear. We see it in parts of the nose. Okay, the thyroid cartilage around the voice box. We see it at the end of the ribs, gives a little bit of flexibility there. We see it in the intervertebral discs of the spinal column. We see it, and we also see it on articulating surfaces, the joints, elbows, knees, hips, places like that. That's where it's restricted to in the adult. So cartilage we consider a specialized connective tissue. It has certain characteristics. Firstly, the amorphous components of extracellular matrix predominate over the fibrous components. So remember, the amorphous components are glycosaminoglycans, proteoglycans, glycoproteins. So they're more abundant than the fibrous components of collagens and elastic fibers. So having said that, it's rich in proteoglycans because the amorphous components are more prominent, and it functions in support and flexibility. Okay, it's kind of bendy, so you can bend things. Also, it's a kind of a good sliding surface. It's kind of slippery, so in articulating joints, elbows, knees, things like that, and acts somewhat as a shock absorber when you're jumping up and down. Your elbows, your knees, things like that can take a bit of a shock without breaking. And interestingly, the presence of cartilage declines from the fetus, where it's much more common, to the adult. And that decline from fetus to adult we see during evolution also, where the early vertebrates had cartilage, and then it's progressively declined and replaced with bone in more developed vertebrates. So the first organisms where we see cartilage are cephalochordates. So these are kind of like fish, but they're not really fish. They're pre-fish. Okay, they're really primitive organisms. Amphioxus is one that's still alive today, and so they're still around. Here's a fossil of one they found in China, 530 million years old. And we see now, for the first time, see cartilage in these cephalochordates. Okay, so that's how far back it goes. If we look at the components of cartilage, we can talk about cellular and extracellular components. So the cellular components consist of perichondrial fibroblasts. So these are fibroblast-like cells at the periphery of the true cartilage that gives rise to the chondroblasts and chondrocytes, which are two different types of cartilage cells. And these cells can secrete the extracellular matrix, which is very prominent. The amorphous component is predominant, mostly glycosaminoglycans and proteoglycans. And we do have fibrous components such as type 2 collagen, which is present in pretty much all cartilage, and two histological types of cartilage, fibrocartilage, for instance in the intervertebral discs where we have now in addition type 1 collagen, and elastic cartilage, like for instance in the hyoid cartilage and places where you have a lot of vibration, epiglottis, where we have some elastic fibers in the cartilage as well. And we're going to go over all of this again. So if we look at the cellular components, at the periphery we have the perichondrium, which is basically the fibroblast-like cells. And these then differentiate into cartilage cells that you recognize as chondroblasts, and as they get older and more mature, eventually they become chondrocytes. Now we distinguish between a chondrocyte, a chondroblast and a chondrocyte, in that a chondrocyte has this territorial matrix, histologically appearing territorial matrix, which is a lighter staining ring, shown here, which the chondroblasts, which are the newer forming cells, which do not have. So in mature cartilage we'll see chondrocytes with this territorial matrix, and then it distinguishes itself from the interterritorial matrix. So these have different compositions of connective tissue. The territorial matrix composition differs to the interterritorial matrix composition. So here's our chondroblast. You can see there's the interterritorial matrix. There's no territorial matrix that we would see of a chondrocyte. Whereas this chondrocyte, now you can make out this territorial matrix around the cell, distinguishing the territorial matrix from the interterritorial matrix. And that's basically it for chondroblasts and chondrocytes. It's really a trivial distinction. Basically, the chondrocyte, which is the older of the cell types, has the territorial matrix, the ring. The chondroblast does not, and that's basically it. We also have cells which are very rare, and you don't read a lot about it, chondroclasts. These are cells that can reabsorb cartilage, and they are related to osteoclasts. And I'll talk a lot more about osteoclasts next week when we talk about bone lecture. But basically, osteoclasts and chondroclasts are derived from monocytes where they fuse together and they have a phagocytic role. So the chondroclasts can reabsorb cartilage to remodel cartilage, okay? So chondrocytes, chondroblasts deposit cartilage, chondroclasts can reabsorb cartilage. And you won't see much about chondroclasts because they're rare to see and there's not a lot known about them. Basically, we think that chondroclasts are similar to osteoclasts. So when we talk about osteoclasts in bone, when you revisit this, when you're looking at this, it'll make sense. Okay. So if you look at cartilage, let's look at the amorphous components. And one of the major amorphous components of cartilage are the proteoglycans, such as hyaluronic acid. So here's the hyaluronic acid. This what appears like a spider web type of material, which covers everything. And it covers over the fibrous type 2 collagen. So you don't see the collagen in cartilage, because even though it's there, because the amorphous proteoglycan is so prominent that it just covers everything. It just acts like a I don't know, a blanket, if you like, that covers everything. And this is what some of these proteoglycans look like. Here's a large hyaluronic acid molecule. It's super high power electron microscopy, and you can see these glycosaminoglycan subunits coming off to form this kind of spider web type arrangement. So it just covers everything and also holds everything together, because you've got all these fibers holding and attaching to different components of connective tissue. So one of the components of connective tissue here is this hyaluronic acid molecule, the glycosaminoglycan, and it's a really large molecule of about 1 million Daltons. It's huge. So you'll see a little bit of hyaluronic acid sprinkled in and around different locations where you have cartilage and other components of connective tissue. So how do we know there are proteoglycans in cartilage? Well, there's this enzyme papain that will dissolve proteoglycans. And so here we have a rabbit that has been injected with papain. So normally his ears would be more erect and the cartilage in his ears would look like this. So after he's been injected with papain, you can see there's a reduced intensity of staining of the cartilage because a lot of the proteoglycans have been broken down in that cartilage, so now it doesn't stain as dark. So this is simply an experimental demonstration, if you like, using the enzyme papain that proteoglycans are prominent in cartilage. Okay, so I talked about the proteoglycans, glycosaminoglycans kind of cover everything, and one of the things that they cover is the fibrous component, which is type 2 collagen. Okay, type 2 collagen is present in all cartilage. There's 3 different types of cartilage. Okay, fibrocartilage, elastic cartilage, hyaline cartilage. Type 2 collagen is present in all of them. Probably the most prominent type of cartilage in all of them. But you don't see it because it's covered over with these glycosaminoglycans and proteoglycans. But it's there, type 2 collagen. And we can stain a chunk of cartilage for type 2 collagen, and we can see from the periphery the fibrous layer, which is depositing the chondroblasts, which are the newly formed components of cartilage, going to the older chondrocytes, which are the older components of cartilage. You can see as you go from the periphery to the center, younger to older, the staining for the type 2 collagen gets darker and darker, okay? Because the cells are older toward the center and they've had more time to deposit the type 2 collagen. Okay, an interesting and highly significant fact about cartilage is that it is non-vascular. There are no blood vessels to speak of in cartilage. Okay, no blood. So that means, what does that mean? That means there's no blood vessels in cartilage, so there's no blood supply. So that means that nutrients and waste products have to make their way into and out of cartilage by diffusion. Now, why don't we want blood vessels in cartilage? Well, if you think of cartilage, it's flexible, right? You can bend it. You know, you've probably had like a bit of chicken breast. You know, the chicken breast, that's a piece of cartilage. You can bend it. You can bend it and release it, bend it and release it. It's flexible. That's one of the functions of it. You can imagine if you had a piece of cartilage and you had a hole drilled right down the middle of it, a blood vessel, and you bend it, it's going to snap right along that hole because that's a fracture plane. So by having blood vessels in cartilage, you're going to eliminate the function of cartilage, which is flexibility. The cartilage would just fracture into a whole bunch of pieces. So then we don't have blood vessels in cartilage to maintain its structural integrity. But because we have no blood vessels, that means diffusion is rate limiting, okay? Diffusion is rate limiting, which means then that the thickness of the cartilage is also limited. You can't have very thick cartilage because diffusion is much slower than if you had a blood vessel penetrating into the cartilage. So if diffusion is rate limiting and the thickness of the cartilage is small, what that also translates into is that if you damage or lose cartilage, it's difficult to regenerate, okay? Particularly as you get older. If you're younger, it can regenerate more so. But if you've ever had cartilage damage to one of your joints, like a knee joint or an elbow joint, where, you know, sometimes they'll get a spur and they scrape it out. Like you see a lot of professional athletes, like football players, they're always going in for some arthroscopic procedure where they're scraping the cartilage out because it's chipped off. Those guys aren't getting their cartilage back, right? And you see stories of professional athletes that are 40 or 50 years old, they're walking around on crutches, right? Football players. And that's because they had these arthroscopic procedures when they were playing football and the cartilage never grew back. So as an older person, they've got damaged cartilage and it's not coming back, and so they've got major problems in old age, okay? So then that's an issue in terms of removing cartilage if it's damaged, especially as an adult. And the reason that the cartilage doesn't grow back is because there's no blood vessels, so diffusion is rate limiting, so you have very limited regeneration of cartilage in adults. Okay, so now let's go back and talk about the perichondrium, which is the fibroblast-like layer at the periphery of the true cartilage. So all cartilage possesses a perichondrium, except for three examples which you need to memorize. The epiphyseal growth plate of elongating bones, fibrocartilage, such as in the intervertebral discs of the spinal column, and articular cartilage in articulating joints, elbows, knees, and so on. That's three locations where you do not have fibrocartilage. Everywhere else where you have cartilage, you do have a perichondrium. Perichondrium basically are these fibroblasts that differentiate into chondroblasts and eventually, as they mature, become chondrocytes. So the perichondrium, being at the periphery, can have blood vessels in it right up next to the true cartilage. So they provide, the perichondrium is a layer that provides via blood vessel nutrients to the forming cartilage. And they are under the control of a transcription factor, SOX9, that then induces differentiation of these perichondrial fibroblasts into chondroblasts and eventually into chondrocytes. And these perichondrial fibroblasts are responsible for what we classify as appositional growth. Basically, there's two types of growth of cartilage. There's the appositional growth, which occurs by differentiation of these perichondrial fibroblasts into chondroblasts and chondrocytes, and interstitial growth, which is simply mitosis of pre-existing chondroblasts and chondrocytes. The interstitial growth is restricted to those locations where you don't have a perichondrium, and I just mentioned them. The articular cartilage, fibrocartilage, and let me see, what was the other one? The epiphyseal growth plate. Okay, so these three locations, the epiphyseal growth plate, fibrocartilage, articular cartilage, cartilage occurs by interstitial mitosis. Everywhere else, you have differentiation of the chondroblasts, of the perichondrial fibroblasts into chondroblasts and chondrocytes. So do you understand that concept? Most of the time you're getting differentiation of this fibroblast-like cell into a chondroblast. In a few locations where you don't have a perichondrium, you get interstitial growth, which is mitosis of pre-existing chondroblasts and chondrocytes. We'll revisit that. But it's pretty straightforward. One's differentiation, the other is mitosis. So there are various hormones and growth factors that will control the growth of cartilage, particularly up until adulthood. Certain steroids, as shown here, and hormones can control cartilage growth. The corticosteroids, sex hormones, can control cartilage growth. Vitamins also, A, can control growth. Vitamin C, we know, can control the growth of cartilage because we need vitamin C for the synthesis of collagen. Remember, type 2 collagen is present in cartilage. So if you had a lack of vitamin C, you wouldn't have good collagen synthesis, so then you wouldn't have good cartilage formation. Okay? And also vitamin D plays a role in calcium uptake, so when you're calcifying cartilage, you need vitamin D for that. So the growth of cartilage is going to be the aggregate of all of these factors, okay? They're all occurring together. Some may be up, some may be down, okay? And the aggregate of all of these vitamins and hormones then will determine the overall growth of the cartilage, whether it's early on, prior to adulthood, or later on, post-pubertal adult growth, which is very little. So now let's go back and look at the two major mechanisms of cartilage growth, the appositional growth, which is by mitosis, and in a moment, the endochondral growth, the interstitial mitosis, I should say. First, appositional growth, which is the most common mechanism. We have our perichondrial fibroblasts that then can be induced via those various hormones, growth factors, and the SOX9 transcription factor to differentiate into chondroblasts, which are now depositing the interterritorial matrix. And then as these chondroblasts continue to grow and get older, they deposit more interterritorial matrix and they push each other away. And then in really mature cartilage, sometimes we see a cluster of, can be chondroblasts or now chondrocytes in what we call an isogenic nest. So this is very mature cartilage, typically in the center of a piece of cartilage. So that's appositional growth. Differentiation of perichondrial fibroblasts into chondroblasts and chondrocytes. Interstitial growth occurs by mitosis. That's the second mechanism of cartilage growth, which is much more restricted. More prominent in fetal and pubertal growth, kind of fades away mostly in the adult. So interstitial mitosis occurs in cartilage that lacks a perichondrium, okay? Lacks a perichondrium. So where would you have a location where you lack a perichondrium? Well, how about an articulating joint, like in the elbow? You could imagine if you had perichondrial fibroblasts and you've got two chunks of cartilage that are constantly moving, those fibroblasts would rub off and slough off, right? Because you've got these two surfaces that are rubbing against each other. So you don't want perichondrial fibroblasts on these two pieces of cartilage that are rubbing against each other, because they're going to get rubbed away in no time and then you've got nothing. So you can't have perichondrial fibroblasts in articulating surfaces, right? You also don't have it in the epiphyseal growth plate shown here, and this will become more meaningful to you when we talk about bone, because the epiphyseal growth plate is a piece of cartilage that grows and is replaced by bone to elongate bones. And the third location is fibrocartilage, for instance, in the intervertebral discs and a few other locations of the body. So these three examples are the locations where typically we see interstitial growth, i.e. the epiphyseal growth plate, shown here, the articular cartilage, shown here, and fibrocartilage, for instance, in the intervertebral discs of the spinal column. These three examples are where we see interstitial mitosis of cartilage. Okay? That's three examples, which is much more restricted. Elsewhere, where we see cartilage, it's going to be by perichondrial differentiation, okay? Interstitial growth, perichondrial differentiation, pardon me. Interstitial growth occurs as a function of growth hormone from the pituitary, stimulating somatomedin C from the liver, makes its way through the vasculature to these locations where it will stimulate proliferation of pre-existing chondroblasts and chondrocytes. So here's an example of interstitial mitosis. We can see the articular cartilage stained in red and the epiphyseal growth plate stained here in red. So these two locations represent two of the three examples of interstitial mitosis, interstitial growth of the cartilage. And this shows you a schematic of different zones of cartilage in the epiphyseal growth plate of an elongating bone. This zone of proliferation is the zone that is responsive to the somatomedin C, then causes these chondroblasts and chondrocytes to proliferate in the epiphyseal growth plate. And we'll revisit this in much greater detail when we talk about bone. Yes? Yes, if you fracture the epiphyseal growth plate, it can impede growth. Like for instance, the epiphyseal growth plate is it's a pretty sensitive type of structure, and it disappears in adults. So as an adult, it doesn't matter because it's gone already. But like during growth, the formation of the epiphyseal growth plate is responsible for the elongation of bones, because the epiphyseal growth plate, the cartilage grows in thickness, but the trailing edge is replaced with bone. So as this occurs, basically you elongate the long bone. People that damage the epiphyseal growth plate, which occurs occasionally if somebody does say there's a fire and they jump out the second story of a building, they land on their feet and they fracture the epiphyseal growth plate in their knee for whatever reason. Okay, if the epiphyseal growth plate is severely damaged, it may no longer be capable of carrying out its normal function. That person may have a stunted growth, a foot, okay? Or so, however you damage the epiphyseal growth plate, if it's severely damaged, it may stunt the growth of an elongation of those bones if it occurs prior to your adult age. And we will revisit that in greater detail when we do bone, because the epiphyseal growth plate is responsible for elongation of long bones next week. Okay, now I talked about interstitial growth in the in those three locations, but also you also get interstitial growth in mature cartilage. So you might have formation of the differentiation of the perichondrium into chondroblasts and chondrocytes, appositional growth, and then eventually in the middle of that cartilage that's formed by appositional growth, you'll get these very mature isogenic nests in the middle of the cartilage. These then can also grow by interstitial growth. Okay, so if you see an isogenic nest, i.e. two or more chondroblasts or chondrocytes together, they may have formed by interstitial growth to form that cartilage. So you can have both mechanisms going on together in cartilage. So for instance, by perichondrial differentiation into chondroblasts and chondrocytes, by appositional growth, eventually as that cartilage matures, right in the middle, if you see clusters of cells like in these isogenic nests, they can also then grow by interstitial mitosis. Okay, so the cartilage then lines the long bones, for instance, in articulating surfaces such as the knees, the elbows, and so on, hips, okay? And sometimes that cartilage can be diseased or damaged, okay? We talked about, like, for instance, during extreme sports, things like that, where a chunk of that cartilage will break off, okay, and it can become lodged in those articulating surfaces, and it can cause pain because you've got a chunk of free-floating cartilage that's getting caught, okay, in your articulating joint. And so that's when a lot of those athletes, they go in and get an arthroscopic procedure where they scope out and remove those chunks of cartilage that are broken. So then in this case, if you have broken cartilage that's coming off the bone, we call that osteoarthritis, relating to the bone that it's attached to, osteoarthritis. There's another form of arthritis called rheumatoid arthritis, which is basically a swelling of the membranes, which pushes pressure on the joints, which is totally unrelated to osteoarthritis other than the fact that it's causing discomfort of the joints. But the two mechanisms are totally different. We will talk more about rheumatoid arthritis next week when I talk about the joint cavities and the joints at the end of one of the bone lectures. Okay, so we'll revisit that. So just remember then that osteoarthritis relates to the damage of the cartilage covering articular surfaces. Okay, we can have certain genetic diseases that can limit cartilage growth. So achondroplasia, A meaning not, if you see A in front of something. Okay, atypia, atypical. Right? A meaning not cartilage growth, defective cartilage growth. So this is common in dwarfism. There are many different mechanisms of dwarfism. One of them is achondroplasia. There are many others. But you can see in this particular individual, he has very short long bones, okay, stunted growth of the long bones. So that has led to his dwarfism. Then the last thing I want to talk about is the classification of cartilage. There are three major histological classifications: hyaline cartilage, elastic cartilage, fibrocartilage. The most common histological form of cartilage is the hyaline cartilage, characterized by the presence of type 2 collagen that's covered over by those proteoglycans. Hyaline cartilage, for the most part, forms by perichondrial differentiation, the appositional growth mechanism. Okay, and you see that in most locations of the body where you have cartilage. In some locations where you have a lot of vibration or movement, you may have elastic cartilage. Elastic cartilage is identical to hyaline cartilage with the addition of elastic fibers in the cartilage. But again, it's formed, you see a presence of a perichondrium, so you have that appositional growth for the formation of elastic cartilage. And you see it in locations where you have a lot of vibration and movement. The histological form of cartilage that is most distinct from the other two is fibrocartilage. Now in fibrocartilage, we now have, in addition to type 2 cartilage, we now have type 1 collagen, which is more fibrous. And we see the cells of fibrocartilage in long parallel rows, which is histologically distinct, which allows us to identify fibrocartilage from either elastic or hyaline cartilage. And because there is no perichondrium in fibrocartilage, it has to occur, fibrocartilage has to form by interstitial mitosis. And it's restricted, fibrocartilage is restricted to just a few locations of the body, such as the intervertebral discs and some other regions of the body. So here's a schematic of our hyaline cartilage with a perichondrium at the periphery. So it's got to be appositional growth. Here's elastic cartilage with a perichondrium at the periphery, so it's got to be appositional growth. And now the elastic cartilage contains the addition of elastic fibers to the interterritorial matrix. But elastic cartilage is pretty much identical to hyaline cartilage with the addition of the elastic fibers. Most distinct, yes? Yes, yes, and fibrocartilage is distinct from the other two because now we have the addition of type 1 cartilage. So hyaline and elastic cartilage will have type 2. Fibrocartilage will have some type 2, but now the addition of type 1. So type 1 cartilage is a distinguishing biochemical feature of fibrocartilage. In addition to the type 1 fibrous collagen molecules, we also have the cells in fibrocartilage arranged in these long chains, as shown here. So here's hyaline cartilage. Again, here you can see the cells and the interterritorial matrix. You really can't see the type 2 collagen fibers there because they're covered over by the proteoglycan molecules. Here's elastic cartilage stained. This purple stain is staining the elastic fibers. So again, you can see a perichondrium, elastic fibers in the interterritorial matrix of this. And here in the epiglottis, which of course vibrates all the time, you can see the blue color is the elastic fibers stained for the elastic cartilage. The most unique is the fibrocartilage. Here you can see the chondrocytes and chondroblasts arranged in these long lines, okay? These long chains following these arrows. And we see fibrocartilage prominently in the intervertebral discs, in particular the annulus fibrosus, shown here. This is the annulus fibrosus. In the middle is a more liquidy, gooey layer of the nucleus pulposus. So here again is fibrocartilage. You can see the cells arranged in these long chains, which is histologically characteristic of fibrocartilage. You won't see cells arranged in long chains like that in either elastic or hyaline cartilage. Again, fibrocartilage, look at how these cells are arranged in these long linear chains. Okay, these chondrocytes, long chains of cells, very characteristic of fibrocartilage. Okay, so here is the intervertebral disc. This is the bone up here. Here's the nucleus pulposus. Here's the annulus fibrosus. In the annuli here, this is where you'd see a lot of the fibrocartilage in the cells. This is a very low power low power light microscopic view, you can't make out distinct cells, but you can make out the annular layer, if you like, kind of like an onion, okay? The layers, and that in those layers is where you will find those cells arranged in those long chains if you were to magnify it. Okay, so sometimes we can have problems with the intervertebral discs, like if you shock, if you jump up and down, you can have a ruptured disc. So the nucleus pulposus is more or less gooey fluid. If you rupture the disc, this fluid can leak out, okay? In which case, the two vertebrae can compress further, okay? And if they can compress further, they might squeeze out the annulus on the side, which can then pinch a spinal nerve, okay? So if you've kind of ever done anything physically, lifted something really heavy, or you've been out jogging or something and you overdid it and you've got some back pain, it could be that you're pinching one of these spinal nerves, as shown here, okay? So that can occur in the intervertebral discs, which of course contain an abundance of fibrocartilage in them. Okay, not so long. So, here's our question. Which type of cartilage is characterized by abundant type 1 collagen? Boy, that couldn't get more timely, could it? So I think we know the answer of that, right? Type 1 is only in fibrocartilage. Okay, 5-minute break, and then shall we do the lab, and then we're done? Yeah? Yes, I'm seeing some nods. Okay, I think we're good. Is it all on? Okay, so let's settle down and we can go through the lab and then we are done for today. So we're going to look at basically connective tissue and cartilage components, see if we can identify things. So here's an example of embryonic connective tissue, pretty much nondescript. Remember, this is the sort of tissue that will differentiate into things like fibroblasts, smooth muscle, things like that. Okay, pretty nondescript, not much else we can say about that. Again, mesenchymal cells looks rather fibroblastic. The point here is that the mesenchymal cells then give rise to many of those other components, of all of the other components of the connective tissue that are not the hematopoietic lineage, okay? The hematopoietic lineage is all of those cells of the blood. Here we're talking about the other components, such as fibroblasts, smooth muscle, endothelial cells, things like that. And another fibroblast, okay? And here you can see it's secreted a lot of material outside of the fibroblast, like this. Okay. Loose connective tissue. So we would classify this as loose connective tissue because you can see how the cells are all pulled away from each other. So this looks like collagen bundles, and here's a few nuclei of fibroblasts and things like that. Some fat cells up here, perhaps. Maybe a blood vessel, fat cell, fat cell, fat cell. So this would be loose connective tissue. Any questions on this one? Okay, and in connective tissue, we can sometimes recognize different cell types at this low power. I wouldn't expect you to be able to identify them, but this is just demonstrating that. For instance, we have some fibroblasts, we have a macrophage, we have a plasma cell that's going to secrete antibodies, and a lymphocyte, okay? So connective tissue outside of an epithelial sheet is going to contain a bunch of different cell types. Remember, we had that big list of cells. of connective tissue, and also the extracellular material secreted by those connective tissue cell types. Any questions? Plasma cells are derived from B lymphocytes. You'll recall the histological features of plasma cells at the light microscopic level is that they have an eccentric nucleus, meaning it's off-center. Here's most of the cytoplasm, so it's eccentric. And quite often the nucleus has this cartwheel arrangement of heterochromatin. So you can maybe this is the axle and these are the spokes, so there's cartwheel arrangement. And of course, the plasma cell, when we see an electron micrograph of it, we can see an abundance of rough endoplasmic reticulum, and of course we have to have an abundance of rough endoplasmic reticulum in plasma cells because they are making immunoglobulins, antibodies, as part of an immune response. And of course, again, this nucleus is eccentric, it's off-center, and you can kind of maybe, if you use your imagination, cartwheel arrangement of the heterochromatin. Any questions on this one? We'll revisit plasma cells when we do the components of the blood. So macrophages, pointing to by the arrow, are phagocytic. You know, we wouldn't be able to identify these as a phagocytic cell type per se at this light microscopic view, but if we happen to have injected an ink into this animal, these macrophages are going to phagocytose up the black Indian ink in this particular case, showing an abundance of phagocytic vesicles in these particular macrophages. So here's one macrophage, here's another, here, here, and so on. Any questions? Okay, adipocytes, fat cells. So this would be a unilocular adipocyte, i.e. single large lipid droplet that fills the entire cytoplasm. The cytoplasm is squeezed to the periphery here. Here's the nucleus, here's a bit of cytoplasm, okay, which would include the plasma membrane, but it's not just a plasma membrane. You wouldn't see a plasma membrane that thick in a light microscopic view. But the lipid droplet is so large, it fills the entire cytoplasm. Any questions? Yes? The nucleus is pointed to by the arrow. See this? The nucleus is pushed to the perimeter. Here is another nucleus here. Here's another nucleus here, okay? Other questions? So here's some more adipocytes stained now with oil red O. The red color, these are again unilocular adipocytes. This is actually a blood vessel here. Here's another blood vessel here. It just gives you a feeling for the size of these cells. So again, these are large lipid droplets that fill the entire fat cell, the unilocular adipocyte. Questions? Yes? How do I know it's unilocular? Because if it was multilocular, there'd be many of these little lipid droplets in there. I know this is a single cell because I know this is a blood vessel, so knowing the structure of the cardiovascular system, I have a gauge in my head as to how big structures are. So these are single cells. And when we do the cardiovascular system, you know, you will become familiar with the size of the various blood vessels. So you'd know that this is a single cell. Other questions? Okay, so here are some more fat cells. These have been prepared for electron microscopy, so they've stained black with, I think, osmium tetroxide. In some cases, the lipid has leached out, so this is just the space, and here's the lipid that's remained inside, stained by osmium tetroxide. Just more views of lipid droplets. Okay, mast cells. Mast cells, similar to basophils in that they mediate an inflammatory response. In other words, they cause localized swelling, for instance, bee sting, mosquito bite, localized edema. The heparin that released from these cells then will interact with adjacent blood vessels, causing them to open up and for fluid to flow out of the blood vessel, and that's where you get the bump, because the fluid has flowed out of the blood vessel into and around the mosquito bite, i.e. localized edema. Edema means fluid retention, so you get a bump, okay? In addition to heparin, in addition to the histamine, which causes the localized edema, there's heparin, which is an anticoagulant, so you don't want things to clog up there. You want fluid to be able to flow into that location. And by opening up the tissue spaces with this fluid, this allows other cells to migrate in, like macrophages to phagocytose the bits of bee sting, and eosinophils to migrate in to mediate the inflammatory response. They typically downregulate the inflammatory response. We will talk about that when we do blood vessels and components of blood. So mast cells mediate an inflammatory response. Okay, they cause localized edema, localized swelling, and they also mediate things like when you have difficulty breathing. What am I thinking of? Yeah, anaphylactic shock is an extreme version. Not bronchitis. When people can't breathe properly, what do you call that? Asthma. Thank you. Had a brain lock on asthma. Yes, asthma is mediated by mast cells and basophils. And in the extreme version, okay, you can have anaphylactic shock where they can't breathe at all, right? So, or for instance, peanut allergies. Some people have peanut allergies, right? Same mechanism. These mast cells are degranulating, releasing these compounds that cause localized edema, or in that case, the whole body becomes swollen up. There's a question there somewhere? I'm not sure, I don't know anything about angioedema. Well, I assume that means edema caused by fluid flowing out of the... So almost like anaphylactic shock maybe? Or, yeah. But yes, any sort of swelling, whatever the result is, it's mast cells and basophils, because they both release heparin and histamines, which facilitate fluid exiting the blood vessels into the tissue space, causing swelling. Question?
Maybe along those lines, but I know some people will react differently to a bee sting. I know my dad would have to have everything around. So what's that? Why would he have such a different reaction than how?
More of the cells being activated. In part, he's more sensitized to it. More cells, faster reaction, something like that. Other questions? And here is a mast cell in an electron micrograph. Okay, so here we can see collagen fibers out here. It's fibers, and here are the granules of the mast cells. So this mast cell has not degranulated. It hasn't released its granules into the connective tissue space yet. It's just sitting there in preparation should something happen. Okay, questions? Yes? Does it always release the same chemicals? Yeah, well, yeah, it's the same chemicals always in mast cells or basophils. It's going to be heparin, it's going to be histamine, right? And then you can also have eosinophil. They're not all listed here. Eosinophil chemotactic factor. You can have glycosaminoglycans and proteoglycans, because actually the glycosaminoglycans also act as a sponge. That's that long linear chain of disaccharide amino-containing sugars like what am I thinking of? One of them, the large one, They act as a sponge. They can absorb a lot of water, so that can also play a role doing localized edema. It will retain the water in the location where you've got this inflammatory response. Hyaluronic acid is what I was thinking of. Yeah, other questions? Okay, so here's a picture of these. So the cells without nuclei are red blood cells. This is a lymphocyte, a monocyte, neutrophils, and platelets. So we're going to revisit this slide and similar slides in great detail when we do the components of blood, hematopoiesis, okay, and stuff like that. Any questions on this one? Okay, so now getting back to classifying connective tissue. Dense, irregular connective tissue would be the dermis. So this light salmon pink layer here is the dermis. Okay. Have we done skin? We haven't done skin. We have done skin. I get confused between Georgetown and here because I'm giving a lot of these lectures. Like tomorrow I'm giving connective tissue at Georgetown, so I kind of lose track. So have we done skin here? Integument? We haven't done integument. Okay, so then you don't, I've given it at Georgetown then. Okay. So integument, you'll learn that this is the epidermis and this is the dermis down here. Okay. So the dermis, the salmon pink layer, is the dense irregular connective tissue, and you can kind of see the orientation, mostly of collagen fibers, in different orientations. Okay, so dermis is a good example of dense irregular connective tissue. Irregular in that the cells and the collagen fibers are all arranged in different ways. Question? Okay, loose connective tissue. So here's the layer of the GI tract, okay, the lining epithelium, a layer, and then down here's the basement membrane, and then here are cells of loose connective tissue, some of the cellular components. We can actually identify some of them even at low power. So this would be a plasma cell. See the eccentric nucleus, cytoplasm, another plasma cell. This looks like a lymphocyte about to differentiate into a plasma cell. Shown here, and maybe some fibroblasts floating around, and a blood vessel. So this would be loose connective tissue if we were to classify that connective tissue. Basically, the lamina propria is the loose connective tissue beneath an epithelial sheet. Any questions? Yes? This would be the basement membrane right here, right? Because we've got this simple columnar layer of cells, right? Simple columnar, bounded at the bottom by the basement membrane. Here you can see the microvilli, very prominent brush border of microvilli. At low power, it looks kind of like uniform, but that's how they look like because they're very densely packed. Simple columnar layer of cells, and at the bottom is going to be the basement membrane right there. You really can't make it out clearly, but it has to be there because you can see that the lamina propria cells now have taken on a different appearance to these simple columnar cells. Questions? Yes? This is a plasma cell here. See the nucleus? It's eccentric to one side. Here's the cytoplasm. Here's another one. Look how eccentric the cytoplasm is to the nucleus. Here's another one. The nucleus and the cytoplasm is eccentric. So that's what I'm calling them plasma cells. I'm based upon the eccentric location of the nucleus, and then you can see the cytoplasm. It looks like it's packed at low power with rough endoplasmic reticulum to make the antibodies. This one, this is a plasma cell, and in part, but the eccentric location is the thing that I'm really going on. You know, the cartwheel thing, that's kind of a matter of judgment, you know. It's kind of iffy, you know. This one's kind of more cartwheel. You got the central axle kind of thing. Maybe this one too, if that's the cytoplasm. This one is not so clear, but the eccentric cytoplasm off to one side is very characteristic of plasma cells. Question here? Not always. Depends upon the epithelial sheet. you might have a basement membrane, you might have some smooth muscle right there, okay? Something like that. But a lot of the time, yes, a lot of the time you will have loose connective tissue to varying degrees. I mean, there's quite a bit here. You might have just a little bit, you know. To varying degrees, you may have loose connective tissue underneath an epithelial sheet, but not always. Other questions? Okay, so here we have some fibroblasts and collagen fibers. So the cellular material here, here's the nucleus, the cytoplasm of a fibroblast. Cytoplasm, cytoplasm, cytoplasm stained darker. And here you can make out the fibrous orientation in longitudinal section of type 1 collagen most likely. And in cross section, you can see the dots here, the punctate appearance in cross section again of the type 1 collagen. Yes? Yes, in fibrocartilage, you only have type 1 collagen. You also have type 2 collagen, but you also have, but type 1 is prominent, yeah. In the pubic symphysis, around the hip area, is another region, and there's one or two others that's listed on that table that I gave you, but that's the other one that I remember, pubic symphysis. Other questions? Okay, so dense irregular connective tissue. Okay, why is it dense irregular? Well, it's densely packed together, and obviously the orientation of what we're seeing is mostly these collagen bundles are irregularly arranged. Some of them are longitudinal, some cross-section, some oblique. So dense irregular connective tissue in this particular case. Any questions? Okay, dense regular. Okay, it's dense because it's packed together very tightly, and obviously the orientation is uniform. They're all running in one plane, so it's a regular orientation. Dense regular connective tissue of the tendon, or we can see similar dense regular orientation and packing in this ligament. So two examples of dense regular connective tissue, very distinct from the dense irregular connective tissue in the previous slide, previous 2 or 3 slides ago. Questions? Okay, reticular fibers. You'll recall reticular fibers stain a blackish color because they are arginophilic. They stain with silver-based stains, and we find them prominently expressed in components of the lymphoid system, so lymph nodes, spleen, thymus, locations like that is where we'll see a lot of reticular fibers, which is type 3 collagen. Any questions? Okay, hyaline cartilage. Hyaline cartilage is, if it's not elastic cartilage or fibrocartilage, then by default it's hyaline cartilage, which is the most common. Hyaline cartilage, in this particular case, we can see a perichondrium, right? Perichondrial fibroblast giving rise to, looks like chondroblasts here, and we can see a territorial matrix around these nuclei, so that would make them a chondrocyte, yeah? Chondrocytes have that annulus that stains differently to the interterritorial matrix. So this is hyaline cartilage with a perichondrium. So this would be appositional growth because these perichondrial fibroblasts differentiated into chondroblasts and chondrocytes. Now, you can have hyaline cartilage in other locations that is formed by interstitial mitosis. So for instance, the cartilage of the articular surfaces of the joints and the knees, that's also hyaline cartilage, but because it's an articular surface, it would have to have formed by interstitial mitosis. Okay, so hyaline cartilage can be both formed by appositional growth and by interstitial mitosis. Here's some more hyaline cartilage, and you can see again the distinct territorial matrix around the nucleus here. So again, these cells that the arrow is pointing to would be a chondrocyte, okay? And because we can see the perichondrial layer here, this would have occurred by appositional growth, differentiation of those perichondrial fibroblasts into chondroblasts and chondrocytes. Questions? Here's some articular cartilage, okay? Note the absence of a perichondrium, right? Because we've got two articulating pieces of cartilage that are rubbing together. If you had perichondrium there, that would just rub right off, okay? So you can't have articular cartilage formed by appositional growth, no perichondrium. It has to be by interstitial mitosis, i.e. these pre-existing chondroblasts and chondrocytes divide by mitosis. Hence, the mechanism of growth of this articular cartilage is interstitial mitosis. Any questions? Okay, again, interstitial growth. If you see an isogenic nest in cartilage, in any cartilage, it can be from anywhere. If you see a nest of cells that are circumscribed as such, most likely these cells, because they're in close proximity to each other and they're enclosed within this nest, formed also by interstitial mitosis. Okay, so you'll see this in mature, much more mature cartilage towards the center, away from the perichondrium. Okay, in very older form of cartilage, you can also get interstitial growth. Any questions? Okay, elastic cartilage. Here we've stained the elastic fibers. These black fibers shown here, specifically stained for elastic fibers. Note the perichondrium. So all elastic cartilage is formed from a perichondrium. Okay, so it's got to be perichondrial differentiation, appositional growth. Okay, questions? And fibrocartilage, our third and most unique and distinct form. Note, you can see the long chains of cells, okay? They're arranged along stress layers, lines of stress. So these long lines of cells and these long chains of cells, that's the histologically unique and defining and distinguishing feature of fibrocartilage as opposed to hyaline or elastic cartilage, okay? Perhaps the easiest, or one of the easiest to identify because of the presence of the cells in long chains. Any questions? Okay, here's an epiphyseal growth plate in a growing long bone. This is all cartilage here. Okay, again, because this is an epiphyseal growth plate, there's no perichondrium. Okay, so if there's no perichondrium, then the cells in this epiphyseal growth plate are proliferating by interstitial mitosis. And this is the mechanism by which long bones elongate up to puberty. We will revisit this in some detail next Monday when we do bone and bone development. Any questions? Okay, electron micrographs of chondroblasts. Why is this a chondroblast and not a chondrocyte? Because there's no territorial matrix, there's no annulus, there's no ring around the cytoplasm. It's very uniform between adjacent cells. So this is a newer formed cell, a chondroblast. As opposed to a chondrocyte, now we can see a distinct ring around this cell, here and here. So then we would say, okay, if this is cartilage, because we can see this ring around the cytoplasm, these would be older cells of the chondrocyte variety. It's a trivial distinction between a blast and a cyte. It's basically that ring around the cytoplasm. Any questions? Okay, that was it. That went pretty quickly. Here's our question. People have problems with this, so I'm going to give you a little time. The image shows an example of one of those five. Okay? What histologically, this could be a lab exam question. You'd see this picture and I'd ask you, what is it? Okay, it's a pretty straightforward question, but people have difficulty with this question. Some people do. Takes a little while to figure it out. Okay, have you all taken your guess in your mind? You've taken your guess? It's hyaline cartilage. Does anyone ask me why it's not one of the other things and why it is hyaline cartilage? Why is it? What if it's not hyaline cartilage, what would it be? Okay, so it's not fibrocartilage. We're not seeing long chains of cells along stress fibers. It's not a plasma cell. We're not seeing rough endoplasmic reticulum. This is the interterritorial matrix. It's not loose connective tissue, all right? Loose connective tissue would have a jumble of different types of cells, plasma cells, connective tissue components. It's not a unilocular adipocyte. These aren't fat cells. You know, they'd be much bigger, right? A big fat cell. Yeah, and you wouldn't see the nucleus in the center. The nucleus would be squeezed out at the perimeter if it was a fat cell. Okay, question? It's irregular. There is no dense irregular, okay? Loose connective tissue is just irregular. There's no dense irregular. There's only dense, regular and irregular. There's no loose, it's all loose irregular. There's no loose regular. That's what I'm trying to say. There's no loose regular. It's all irregular because it's loose, right? It's loose, so it's flopping around. There's no loose regular. It's only loose irregular. But there's two types of dense, regular and irregular, okay? Yes? Yeah, this is a forming isogenic nest. These two cells together, it looks like a forming isogenic nest down here. See, these two cells look like one of them gave rise to the other, all right? So yes, we could say this is an isogenic nest. There are much better examples of isogenic nests where you might have 4 or 5 cells together. But yeah, like this, that's an isogenic nest, and it looks like it's got a ring of something stained around it differently. Here, an isogenic nest forming here. Other questions? Yes? Yes. You'll always see an image, and then it's a multiple choice question. So you will have an image projected on the screen, and in your hand you will have a written printout of the question A, B, C, D, E. The questions aren't next to the image. The question is on your, and it'll be question 3. And you have 60 seconds to answer it, okay? And I'll say, and then at the end we say, okay, next question. So that's your chance to write something. You can, and you know, the exam itself, you can write on there maybe A or maybe C, and you can go back at the end if you need to time to think about it. Yeah, question? Not that I'm aware of. Other questions? So the question was, are there any other cells that have isogenic nests for the recording? Not that I'm aware of. Okay, so no more questions. So on Monday, we're going to continue with connective tissue, and we're going to look at bone and bone development, and we're going to see how there's a continuum in the evolution of connective tissue components, in the embryological development of connective tissue components, and how those two have significance in terms of the sequence of events during bone repair when you break a bone. Okay, guys, enjoy the weekend, I guess, in a couple of days, and see you next week.